Welcome to the NeLS portal. In this tutorial I will explain you how to transfer data from NeLS portal to the StoreBioInfo and how to transfer data from StoreBioInfo to the NeLS portal. So once you log in on the NeLS portal, by default you will be under home and under my data, and where you can see the personal folder, and if you click my project you can see your projects. So to transfer the data from the NeLS to the StoreBioInfo, there is one option available in the NeLS. If you see this one, if you click here StoreBioInfo, this is the area or the option available where you can transfer files from NeLS portal to StoreBioInfo and from StoreBioInfo to the NeLS portal. So first I will tell you how to transfer the file from StoreBioInfo to the NeLS. Now I have the personal folder which is available under my data, and I have the projects which is available under my project. So before we start transferring the files, let's have a look of the directory or the folder structure here on the NeLS. So this area personal, if I click here, this area personal is available under my data. So if I click here, I have the personal area, that is my area that I can keep my files or manage my files. And if I click the StoreBioInfo again, and then there is another area or another folder, projects. So if I click the project, I will be inside the project, and oops, sorry, the slow internet connection. So I will be inside the project, and this is the area, this is the same area which is available under my project. So if I click here the my project, and I can see this project, so this is the same area with which I can see under StoreBioInfo. So if I click the StoreBioInfo, I can see personal, that is here available under my data, and the projects, that is available under my project. Now on the other side, the StoreBioInfo directory structure. Inside the StoreBioInfo, I have one project. You can have one project or you can have multiple projects, and inside the one project, if you click here, inside one project you will have the one data set or multiple data set, and once you click the data set, you will have the sub data type, reads and analysis. So you can transfer the data from the reads or from the analysis, if you click here, to your NeLS portal. The important thing here to understand is that StoreBioInfo has a structured file system. So you cannot copy data anywhere, you cannot paste the data or you cannot transfer the data anywhere inside the StoreBioInfo. You can only transfer inside the analysis or inside the reads. So that means you can only transfer two types of data. The first one is the reads, that is your input or the raw files. In my case, I have the FASTQ files. The second type of data that you can transfer to the StoreBioInfo is the analyzed data, that is your intermediate or final results after the analysis. So you can only transfer two type of data. I will repeat. The first is your raw data. In my case, it is the FASTQ file from the Illumina sequencing run, and the second one is the analyzed data, that is intermediate or the analyzed data after the analysis of my FASTQ files. So two type of data I can transfer here in the corresponding sub data type. Now but on the NeLS portal I can transfer these kind of data, either reads or the analyzed data, anywhere inside the personal folder or in the correct subtype under my project. So first I will show you how to transfer the data from StoreBioInfo to the NeLS portal. So if you click here StoreBioInfo, and now I want to transfer from the StoreBioInfo to the NeLS portal. So I will click my project, and then I will click my dataset to go inside, and now I want to transfer one FASTQ file from here to my personal folder. So I will click the personal, and now I will select the file that I want to transfer, and I will click this option, transfer to the NeLS. So the thing to remember is that I am inside the project and inside one data set and inside the reads, that is a subdata type, and then I am inside the personal folder and the place where I want to transfer my files, and after that I have selected the file here by clicking here, and then I will click here, and then it will start transferring my file. So if I click here, transfer to NeLS, and there is a message, you have initiated the action successfully. That's all. That means my data is being transferred to here, and depending on the file size it will take some time, and then I will be able to see my data. So my data, this test1 r1 FASTQ file is 756.2 KB, and once I click here I can see one file 756.2 KB. That file is successfully transferred here. So this is the way I can transfer the data from StoreBioInfo to the NeLS portal. Now I will show you how to transfer the data from the NeLS to the StoreBioInfo. Now I will select the target that I want to transfer to the StoreBioInfo. So I will go under the project, will click here, and then there is one project, test project. I will click here, and then there is one data set, sample1. I will click here, and inside here I have the reads and I have the analysis folder. So I want to transfer some reads file here. So I will click here, and I want to transfer this file into the StoreBioInfo. So here the important thing is to remember that I cannot transfer anything directly from here to the StoreBioInfo. I can only transfer my data in the correct subdata type. So what does it mean? It means that I should go inside the test project, and then I should go inside the data, and then I should go inside the correct subdata type, and then I can transfer here. So if I select the file here and then click the transfer, and then it will start transferring the file. Now before that I want to show you something, some important thing. Once I have selected the data, I have one target data. This data I want to transfer to the StoreBioInfo. So I will select the test for project, then I will select the data set, but here the important thing is to remember is you see the status, logged status is false. So once the logged status is false, this means I am ready to transfer any data to this data set, and once this logged status is true, that means I cannot transfer any data to here, and that true status will be available once the process is going on inside this data set. So how? I will show you. Now I have one target cancer2 underscore 1 FASTQ file that I want to transfer here, but I cannot transfer anywhere. So I have to select the correct data set. I want to transfer in this data set. I will click here, and then I have to select the correct sub data set. So I want to transfer inside the reads because these are my reads. Now I am ready. So I will select the file, and I want to transfer from here to here, just selecting file and click transfer to the SPI. Once I click this one, you will see the message. You see that is saying you have initiated the action successfully. This data set and all sub data types have now been logged until this file transfer is finished. Always inspect that your data has been successfully transferred before deleting any original data. Now what does it mean? It means that it's saying this data set and all sub data type. What is my data set? Here is my data set. So this data set is locked. And why it is locked? Because the file transfer is going on. Once the file transfer will complete, my data set will be unlocked again. So I want to see whether it is locked or not. You see, the locked status is true. This means now I cannot go inside the dataset and I cannot paste anything inside the dataset. This dataset is locked. So let me try. If I click here, I cannot go inside the dataset because this is locked. But once the file transfer will complete, and it will take some time depending on your file, and once the file transfer will complete, my data set will be unlocked again and the status here will be false. And once the status here is false, I can transfer, I can select one file or another data set, and I can start transferring again. This is the normal procedure. Second thing is that I already also had an option that I can select the multiple files here, or by just selecting here it will select all the multiple files, and these all files I can transfer directly to the I can transfer these all the files or the data set into the reads inside the reads. So in just one click I can transfer all these 3 files or 5 or 10 files inside my reads. So I don't have, in that way I will not have to click or transfer single file. I can do it in just one click, and the status will be the same, true. It will be logged unless and until the file transfer is complete, and then it will be the false again. So and then I will be able to transfer the file. So I hope it is clear, but if you still have some question or if you are confused, you can watch the other tutorials how to ask the help.